So you'll recall that we talked about four major group of eukaryotes, all belonging to domain Eukarya. Those were the protists, and we said protists were eukaryotes. Many of them are unicellular microbial, and they're neither members of kingdom Fungi, nor members of kingdom Plantae, nor members of kingdom Animalia. And folks, with this I have to apologize because there's been, at least in my mind, some debate or confusion over the classification of Sprirogyra. So although Sprirogyra can be classified as an algae, a green algae, it is part of kingdom Plantae. It belongs to a primitive group of kingdom Plantae. So even though in many of our references, like our lab manuals and the older PowerPoints and older notes, we say it's a plant like Protist, officially it's part of kingdom Plantae. Because of that confusion, I won't ask you if Sprirogyra belongs to kingdom Plantae. I won't ask you is Sprirogyra a Protist. I just want you to know that it is a eukaryote, and we'll see that it has beautiful green chloroplasts, so it can carry out oxygenic photosynthesis. Okay, so starting out with apologies is not good. So folks, we will be seeing then as an example of animal-like protists. We'll be looking at the an amoeba, Amoeba proteus, which is a free-living little protozoan. We'll then be looking at members of kingdom fungi. We'll look at unicellular fungi called yeasts. We'll be looking at Saccharomyces cerevisiae, also known as baker's or brewer's yeast. And then we'll be looking at the filamentous mold, Rhizopus, also known as bread mold. Well, then our example, folks, of members of kingdom Plantae, we would go back to our microscope lab. Remember the aquatic plant Elodea. And again, we will have slides of Sprirogyra so we can see their beautiful green spiral chloroplasts. And we'll also see that Sprirogyra can carry out both sexual and asexual reproduction. Then we'll finish with kingdom Animalia. Of course, Homo sapiens, humans, we are members of the animal kingdom. And then we'll look at a larval form of a parasitic worm called Taenia solium, the pork tapeworm. We'll be looking at little cysticerci, resting forms. And then we'll end with the mosquito Aedes aegypti. And we'll discuss the bad news that they've recently found breeding colonies of Aedes aegypti here in Northern California. And that's of concern because Aedes aegypti are vectors for different viruses, yellow fever virus, Zika virus, dengue virus, chikungunya virus. So we'll talk a little bit more about that later. So first, folks, you'll recall that at one time there was a kingdom called Kingdom Protista, and this was kind of a dumping ground for all the eukaryotes that weren't fungi nor plants nor animals. And now with DNA and RNA sequencing, they've shown it's not a kingdom because the members are so diverse. But you'll recall historically, biologists tried to make some sense out of these protists by describing them as animal-like protists or protozoa, or plant-like protists, algae, or fungal-like protists such as slime molds and water molds. So we're going to look at a animal-like protist, the protozoa Amoeba proteus. And again, folks, for the exam, you want to know classification. So Amoeba proteus, it's a eukaryote. It belongs to domain Eukarya. It belongs to that broad group, the protists. It's an animal-like protist, a protozoa, and some of the features that remind us of animal cells is that it lacks a cell wall. So remember, all animal cells lack a cell wall. It does have a cell membrane, a cytoplasmic membrane. It's a chemoheterotroph, which means it has to have preformed organic molecules. It's a source of energy and carbon. It has an ingestive form of nutrition, and it's really cool. They'll form these cytoplasmic extensions called pseudopods or pseudopodia, false feet, and with them they'll surround a food particle, maybe like a bacterium. The pseudopods will fuse, forming a food vacuole, a phagosome, and then with the fusion of lysosome hydrolytic digestive enzymes are dumped into the phagolysosome, and the little food or the bacterium would be digested. So again, they're animal-like in their chemoheterotrophs. They have an ingestive form of nutrition where they bring the food into their cells to digest it. And another animal-like property is they're motile, and these pseudopods have a second function. They help amoeba move over solid surfaces. So, and that's very similar to animals. In some of your slide boxes, the company that prepared the amoeba proteus slides for us, they got a little carried away. What they did was they took 3 different tubes of amoeba and added 3 different dyes, and then they mixed them all up and created their amoeba smears. So some of you, the amoeba look like fireworks. They're pink and turquoise and yellow. So just recognize, you guys, that in nature, amoeba are usually colorless. We'll next move to kingdom fungi. In kingdom fungi, we have unicellular fungi, the yeast, and then we have multicellular filamentous fungi called molds or fleshy fungi. So we're first going to take a look at the unicellular yeast called Saccharomyces. So again, for classification on the lab exam, you guys, it's a eukaryote, unicellular, domain Eukarya, kingdom fungi. Because it's a unicellular fungus, we call it a yeast. And what's really cool with yeast is they can reproduce asexually by a process called budding. So we're hoping that we'll have a fresh culture of Saccharomyces. We'll mix some Saccharomyces in some water, give them a little bit of sugar, and warm them up. And they should start reproducing asexually by this cool process called budding. So in budding, if we watched one cell, it would appear as if the little cell is growing almost like a little nose, and that's the bud. And the bud grows bigger and eventually separates from a mother cell, and then it too can grow and divide. So we see lots of budding going on here. There's a bud, there's a bud, there's a bud. And again, hopefully that's a beautiful bud. We'll see some budding in live cultures in the lab. Saccharomyces cerevisiae is also named baker's or brewer's yeast. And the reason it's used in baking leavened bread or in making beer or wine is that these little guys carry out this cool process called alcoholic fermentation. What they'll do is they'll take glucose and they'll oxidize the glucose, and then we'll have end products of carbon dioxide and a 2-carbon alcohol called ethanol. So this is the alcohol. And in the process, per glucose molecule, the little yeast will benefit by having a net gain of 2 ATP. So again, the Saccharomyces is used in bread, beer, and wine production. Oh, and we might wonder why when we eat bread we're not getting alcohol. And it's because when we bake the bread, the heat causes the ethanol to evaporate. So here's our baker's yeast, and here's some beer, fermentation product usually made from grains with the help of Saccharomyces. The next member of kingdom fungi will be the multicellular filamentous fungus, which we call a mold, Rhizopus. And Rhizopus is often called bread mold. So again, classification, domain Eukarya, kingdom fungi, multicellular filamentous mold. Again, Rhizopus called bread mold. And these filaments are actually chains of cells. And these long filaments, thread-like structures, they have a fancy name. They're called hypha. And fungi are really cool. Many of them can reproduce both asexually and sexually. So we'll see on some of the molds that we study, we can see formation of asexual spores, and in Rhizopus, asexual spores are formed in these little balloon-like structures, and the balloon-like structure is called a sporangium. It's supported by what's called an aerial hyphae, a hyphae that grows up into the air. And within the sporangium, asexual sporangiospores are formed. When they're mature, the sporangium breaks open, and then the spores will be spread by little air currents. They'll be spread to new locations, new sources of food. And then furthermore, we'll see with Rhizopus, they can also reproduce sexually. That means we're going to have 2 different fungal parents, genetically unique. They'll sense one another by the presence of chemical pheromones, and as a consequence, the two parents will grow these little lateral extensions here and here. And when the two parents meet, they're going to combine their genetic information, forming a zygosporangium or zygospore. So on a lab exam, you can use either term. And the important thing to remember is that the zygospore is a product of sexual reproduction. And this is really important because it helps increase genetic diversity within populations of Rhizopus. And we know that's really important for natural selection. So folks, this would just be really just a flashback to our microscope lab when we looked at Elodea. So you guys help me out here. If this was a lab exam, let's pretend this is a lab exam. So we're looking at Elodea. Which domain does it belong to? Which kingdom does it belong to? What are these little oval green structures? What's the function of the green structures? And why are they green? So just really quickly, the answers, you guys. So Elodea belongs to domain Eukarya, kingdom Plantae. The green structures are chloroplasts. The function of the chloroplast is oxygenic photosynthesis. The reason they're green is from that really cool photosynthetic pigment, chlorophyll A. And furthermore, you guys remember, eukaryotic chloroplasts evolved from primitive endosymbiotic cyanobacteria according to the endosymbiotic theory. And again, you guys, Sprirogyra, oh, and I misspelled it. That's so not good. Sprirogyra, in the old days, I would have, it's not going to let me change it. In the old days, it's not letting me change it because I'm doing a movie. Okay, you guys, I'll have to correct it later, sorry. So Sprirogyra should be spelled S-P-I-R-O-G-Y-R-A. That first R shouldn't be there. So previously, we included Sprirogyra with the protist, the plant-like protist, the algae. But again, looking into the classification, the Sprirogyra are actually included in kingdom Plantae. And I think the reason I got confused is I always think of members of kingdom Plantae looking like, you know, trees or bushes or flowers. But there's multiple subgroups of kingdom Plantae, and one of the groups are of these really primitive plant-like organisms. Primitive in that Sprirogyra don't have differentiated roots or stems or leaves. Yeah, and indeed, folks, we still call it a green algae, but again, it's now classified in kingdom Plantae. I would never ask you on our lab exam which kingdom Sprirogyra belongs to, right? Because it is so confusing. But it would be fair, folks, for me to ask you the following questions here. So, is it a prokaryote or a eukaryote? It's a eukaryote, right? It's going to form these chains of beautiful cells. So this is how it appear in nature. And really neat, they have these beautiful spiral chloroplasts. That's where the name Spiro comes from, these spiral chloroplasts with chlorophyll A, and the chloroplasts carry out oxygenic photosynthesis. Okay. And the Sprirogyra can reproduce both asexually or sexually, right? And you might, on some of your slides, you might see stained slides with Sprirogyra reproducing sexually, and they're pretty cool. So in those slides showing Sprirogyra undergoing, this process is called conjugation. We have two parents. This would be like one parent, maybe the equivalent of dad. This would be the other parent, the equivalent of mom. And what you'll see is when they exchange chemical messengers, pheromones, they'll grow these lateral extensions, excuse me. And then the one parent will pass the equivalent of the male gamete through this little conjugation tube, and the male gamete will fertilize the female gamete. So what we're seeing here is the result of that fertilization, these really cool zygotes. And again, folks, sexual reproduction, it's such an advantage because in this way, populations can really increase their genetic diversity. And that, again, is going to be really important for natural selection and biological evolution. Kingdom Animalia, we want to remember that we're members of Kingdom Animalia. The other examples of Kingdom Animalia, we'll be looking at a resting stage, an immature stage called the Cysticercus, of a parasitic worm called the pork tapeworm. And this is an example of a mature pork tapeworm. They can be multiple feet long. The adults live attached to the inside of our intestinal wall by their head, which is called the scolex. And the head is pretty cool, you guys. It's got hooks and suckers. And then there in the food-rich intestine, the tapeworm starts growing these little segments called proglottids. And it really does look like a tape measure you use in carpentry or in sewing. And really the main purpose of the proglottids, each proglottid is an egg-producing factory. So the most mature proglottids, this is, you can barely see the head up here. This is the tail end. These are the oldest proglottids. When the proglottids are mature, they're going to be chock full of eggs, and this proglottid will break off and get passed in the feces. And then when the proglottid dries up, it looks like little rice grains. It breaks open, releasing the eggs. So the eggs will be in the feces. So wherever the feces goes, the eggs go. So if the feces contaminates food, or if it contaminates water, and then an animal or a human ingests those eggs, then we have another part of the life cycle. So we'll follow it through, you guys, just using pigs here. So where's our come on, little pigs, down here in the lower right-hand corner. So here we have a pig that's ingesting eggs, and this happens sometimes when you have free-roaming pigs, and maybe there's human feces contaminating garbage, and the human feces can have these eggs in it. So that the pig eats the eggs. The eggs, when they enter the pig's intestine, the eggs hatch, releasing these little baby larvae, little larval immature worms. They penetrate pig's intestine, and then they travel throughout the pig's body. And once they reach a good location, like maybe say the muscle of the pig or the tongue of the pig, they'll form these little resting structures. This resting structure is called the cysticercus, and it's basically just a little immature worm that's waiting to be eaten, right? So if we're a human and we eat pork and we don't cook the pork thoroughly, we won't kill the cysticerci. And then when we swallow the cysticerci, they'll emerge in our intestines. There's a little head here. You'll even get to see the hooks possibly in your microscopes. The cysticerci, they emerge, they attach to our intestinal wall. By the proglottids start growing, so they will end up with adult tapeworms in our intestine. Of huge concern is if humans swallow the eggs. So again, this could be through fecal contaminated food or water, because the same thing that happened in the pig is going to happen in us humans. The eggs will hatch in the intestine, releasing baby worms. Baby worms penetrate the intestine, spread throughout the body. And for some reason in humans, the little baby worms love the brain. And so they'll form all these little cysticerci in our brain. And here's an autopsy from a person that probably died of a disease called neurocysticercosis. And all these little holes, it almost looks like Swiss cheese. All these little holes are where the cysticerci, the immature Taenia solium, have formed. And of course that causes massive brain damage here, right? And so if a person is continually swallowing eggs because they don't have access to clean water or food, they can have lots and lots of brain damage from neurocysticercosis. And you will see cases here in Northern California. One student, I think about a year ago, said that they had a patient present with neurocysticercosis. And I know that a few years ago there was another case presented at the UCD Med Center. So you will see patients, suffering with neurocysticercosis. In one of the YouTubes that I think I had a link for, it said neurocysticercosis is one of the most common causes of epilepsy worldwide. I had no idea. That is just crazy. And again, here, folks, from one of the YouTubes, it said there was 50 million cases worldwide, so it's pretty common. And this is just some facts, you guys, from the World Health Organization. Again, I had no idea that worldwide this was such a huge problem. So here we have free roaming pigs, and they have access to a garbage dump. And the concern would be here, well, actually there's two concerns here, and that is if there's human feces here that has the Taenia solium eggs in it, then the pigs would end up with cysticerci in their tissues, right? And then if we eat the pork, the pig tissue with the cysticerci without cooking it thoroughly, then we can end up with adults in our intestines. And again, if we have adults in our intestines, we might have fecal contamination of our fingers or something, and by accident maybe swallow those eggs, right? And then our feces, of course, would be a source of eggs for other people. So this is not good. Yeah, here it is, you guys. Neurocysticercosis is the most prevalent and preventable cause of epilepsy worldwide. And it makes sense, you guys. It's mostly going to affect rural farming communities. And often those are the communities that have the least access to good medical care. Taenia solium was recently labeled the foodborne parasite of greatest global concern by WHO, the World Health Organization, and FAO, the Food and Agricultural Organization. So this is really bad news. And again, folks, let's see if I can barely read that. This is endemic. Endemic means it's always present. So the prevalence of neurocysticercosis, endemic, always being transmitted here, which suggests probably there's pig populations here that are infected. So parts of South America, Central America, up into Mexico, some parts of Africa, some parts of India, up into China. So if you have patients that present with neurological signs, maybe epilepsy, it's really important to know where have they lived or traveled, and very importantly, do they eat pork, right? This might be one reason why in many traditions eating pork is forbidden. It might be that even before folks knew about parasites, that some cultures saw a connection between the ingestion of pork and maybe later some, disease. So, and again, it's really interesting that pork is not eaten by many cultures. And this is our last animal, folks, mosquitoes. I was reading an article and it said mosquitoes are the most lethal, deadly animals in the world. And you're kind of like, what? They do drink our blood, but they don't drink that much blood. The reason they're the most lethal or deadly animals in the world is that they are incredible vectors, arthropod vectors, for microbial pathogens. And this is a photomicrograph, not a photomicrograph. Mosquitoes are macroscopic. We don't need a microscope to see them. This is a female Aedes aegypti, the yellow fever mosquito. And the reason I know she's a female is in mosquitoes, only females are the blood feeders. I thought that was amazing. So the males are important pollinators out there in nature. So the males don't drink blood. And you might think, well, why do the females? Well, the females have to have lots of rich food to make all of their eggs. And that's why the females are the blood feeders. They need rich blood so they can make their eggs. So Aedes aegypti, you guys, again, I think I mentioned earlier, just this summer, breeding populations of Aedes aegypti were found here in Northern California. So this is bad news because they can vector many different types of viruses, the yellow fever virus, Zika virus, dengue virus, chikungunya. And with Zika virus, Zika has received a lot of press here lately because what makes Zika virus infections, what should I say, even more serious, is that if you have a pregnant mom and she gets infected with Zika, the Zika virus can cross the placenta. We call that transplacental transfer and invade the developing baby. And Zika virus loves to invade, replicate in the baby's neurons. And as a consequence, then, the baby ends up with really serious neurological damage. In really serious cases, when the baby's born, the baby will have what's called microcephaly, a small head, and it's because the brain hasn't grown properly. But even in babies that are born with a normal size head, and if mom was infected with Zika during pregnancy, it's possible the baby still will have neurological damage. So this is just such bad news, you guys. Right, to have Aedes aegypti here in Northern California. They're aggressive daytime biters, right? So usually we think of mosquitoes at night. These guys come out during the day, and they're very aggressive. But one thing we can do, you guys, this really can be helpful. Check around your house. If there's any containers with standing water in them, dump them out. And the reason is female mosquitoes have to lay their eggs in water. And the eggs have to hatch in the water, releasing the little immature forms. And these little immature mosquito larvae and pupa, they have to develop in water. So if we can decrease the amount of standing raw water near our homes, then we can decrease the breeding grounds for the mosquitoes. We can keep the mosquito numbers low. And another thing we can do, folks, if we have maybe little ponds, and if you have a little pond, maybe for aquatic plants outside, and you don't have fish that will eat the mosquito eggs or mosquito larvae or pupa, the Sacramento and YOLO mosquito and vector abatement and control organization will give you free mosquito fish. They're called gambusia. So I'll try and remember to bring some to lab because they're really cool. We've got them. We've got a pretty good, healthy mosquito fish population growing in our backyard in a little pond. So I'll bring some in. And if some of you might need some mosquito fish, I can share. I can share some of them with you. Okay. All right, you guys. I think that ends our lab unit 6, cast of characters. And folks, one thing that might be kind of cool is that this PowerPoint might actually help you with our first lecture exam, because you recall in lecture in the first unit, we did a unit on the cast of characters, so maybe this PowerPoint will help reinforce some of those concepts. Okay, folks, so let me see if I can get out of here.